Good evening, everyone. I hope I'm audible. If I'm audible, you can respond yes or no in the chat box. Okay, so I'll start the session. Looks like more people are joining in. So I'll wait for just 2 more minutes and then I'll start the session. So I'll start the session now. Good evening, everyone. I'm Pushpangna, and I'm a PMRF scholar from IIT Kanpur. And today I'll be taking this first problem-solving session for the NPTEL course Cell Culture Technologies, which is taken by Professor Manak Das. So I'll be basically dealing with the week one questions. So the assignment questions which you get for every week will basically be dealing with them so that you have a fair idea of how to solve them and what the answer to those questions is going to be. So we can start with the first question. The first question is primary culture becomes what after the first subculture? So there are four options. The first is cell system, the second is cell line, the third option is stem cells, and the fourth option is cancerous cells. So, if you have listened to the NPTEL lecture videos by Professor Manak Das carefully, you'll know that this is a very this forms primary culture and cell line forms the basic of the cell culture technology course. So, primary culture after its first subculture becomes a cell line. So here the correct option will be B. So your primary culture will become a cell line after its first subculture. So I'll just explain it in brief, like what basically is a primary culture. So what happens is you have your organism. So from the tissue or organ of that organism, you take out some cells. Suppose you take out some cells, and then you decide to grow or to culture them outside that organism. So here, whatever is happening in this organism system will be referred to as in vivo. But if you take out the cells and then you decide to culture those cells outside that organism, not inside that organism, then it will be something which will be referred to as in vitro.
Ma'am,
Yes. Hello. Someone wants to say
ম্যাম
something.
শোনা যাচ্ছে না কিছু?
Hello.
ম্যাম শোনা যাচ্ছে না কিছু?
Can you please speak in English? Hello.
ma'am, you are not audible.
Okay. Okay. Hello.
Hello, ma'am. You were not audible from
Oh,
starting.
okay, okay. I'm so sorry. I'll start again. Okay. So I hope I'm audible now.
Yes, ম্যাম.
Yes,
Yeah.
ma'am.
So I was just discussing the first question. So I'll basically be dealing with the questions which are there in your week one assignment, And I'll just give you an idea of how to solve those questions so you can submit those assignments yourself. So basically the first question is primary culture becomes what after the first subculture. So there are four options. The first option is cell system, the second option is cell line, the third option is stem cells, and the fourth option is cancer cells. So if you have paid attention to the video lectures by Professor Manak Das, you will know that primary culture, cell system, cell line, these form the basic of like all the basis of your cell culture technology. So what happens is primary culture becomes cell line after the first subculture. So here the correct answer is the option B. So I'll explain it in brief. Why is it so? What is primary culture and what is cell line, and how from a primary culture we get a cell line. So here what happens is, suppose if you are taking out some cells from some organism or from the tissue or organ of an organism, and then you decide to culture only those cells outside that organism. So inside that organism system will be known as in vivo. And if you plan to culture those cells, like you take out those living cells and you plan to culture them outside the organism, this system will be called in vitro. So your cell culture is basically an in vitro technology. So here, when you are taking out these cells directly from the organism, this is something which is called a primary culture. Primary means the very first culture that you are taking out directly from the organism. Now, once you have taken out these cells and you decide to culture them in an artificial environment in your laboratory in petri dishes, then like once you go on subculturing your primary culture, this gives rise to a cell line. So here, growing the very first set of cells derived from the tissue of an organism, living organism, is your primary culture. And when you subculture or passage it, so these are the two terms which are used very routinely in cell culture, that is subculture or passaging. What does it mean is that you are continuously, from the existing cells, you are continuously regrowing those cells. So for your primary culture, when you develop a cell line, this will be your passage, first passage or first subculture, and then from there you can go on passaging them many times, like for n number of times. Now moving on to the second question. Second question is about confluency. So here the question is 80% confluence means, again we have four options. The first is cells have a size of more than 80 nanometer. Second option is 80% of the cells have died. The third option is 80% of the cells have divided. And the fourth option is cells have covered 80% of the substrate. So before I discuss the answer to this question, I'll show you an image where you can see like there are different percentages of confluency of cells. So here, as you can see, they are only 10% confluent. Here they are 20% confluent. Here they are 30% confluent, and so on. Here you are seeing that they are 100% confluent. Now, if you compare this image of 10% confluency and this last image of 100% confluency, what you will see is that here there are very less number of cells. But here, like these cells have completely covered the surface. So confluency basically means like what percentage of the surface of the substrate has been covered by the cells which you are culturing. So here the correct answer will be your option D, that 80%, if we are saying that our cells are 80% confluent, that means cells have covered 80% of the substrate, and that is clearly visible if you compare the different confluency percentages in these images. So the third question is, cell lines that can divide a particular number of times until they reach something are called what? So again, there are four options. Now here in this question, it is mentioned that we are talking about only those cell lines which can divide a particular number of times. That means, uh, like the number of times that they can divide is finite. Like there is a fixed number n. It's not like uh, they are going to divide indefinitely or they are completely immortal. So like cells which are going to divide for a finite number of times will be called finite cell lines because they are not immortal, they are not continuously dividing. So these finite cell lines will divide for the n number or the for finite number of times until and unless they reach something which is called senescence. Senescence basically means like old age. So here we are talking about the age of cells. So once they start to lose vigor, they start to lose potency, these finite cell lines stop dividing. So here the correct answer will be option A. Now again, I like to discuss what are finite cell lines and what are infinite cell lines. So here again, so the first source of our cells for starting any cell culture or for starting any cell line is going to be your organ or your tissue. So this is your primary culture as we discussed in the very first question. So this is the primary culture. So from the primary culture, like from the organ, you are going to take some cells, you are going to isolate those cells, and from here you are going to culture those cells. So like suppose you took only 4 5 cells from the organ, and then when you culture them, it like with due time, with a good media, with good serum, it will become confluent. Like here you can see that it is around 90 to 95% confluent. So once you have a confluent culture, that means once your primary culture cells which have been subcultured have covered the entire substrate or the surface, then again what you can do, you can go on subculturing them because you don't want to just stop at one stage. You have to like culture those cells further as per your experiment, as per your need. So from here, when you do secondary culture, then you get your first cell line. So once you get your first cell line, like the very cell line which is derived from the primary culture, so this will be your primary cell line. Your primary cell line, when you go on subculturing, then it will be your secondary cell line and so on. Basically, these are all concepts of when you're going to passage or subculture your cell. So from here, what you're going to do, like you're going to continuously subculture your cell. But what will happen is, it's not like you can go on subculturing your cell indefinitely. So what is happening here is, cell division is taking place, but it is taking place only till only for a finite number of divisions. It's not going to keep happening. It's not immortal. Now, so these are finite cell lines. These are so most of the like cells that we routinely use in lab are actually finite cell lines. But what happens is, imagine a different scenario where again you are going to take a primary culture, but this time you are going to take it from an organ which has a tumor. Tumor means it has cancer. So, like when you are going to like isolate cells from an organ which is not a normal organ, it has a tumor, and then when you are going to culture those cells, then what will you get? You will get transformed cells. You will get transformed cells. You will not get the same set of these black normal cells. So here, what will happen is, if you are taking cancerous cells and then you are culturing them and then subsequently subculturing them, so they have this property that they are immortal. They can go on dividing indefinitely, and so these kind of cancerous cell lines will be your continuous cell line. So these cell lines you can go on subculturing to indefinite number of divisions because they are not normal cells, and that is why they do not have an age. Whereas your finite cell lines will have a senescence period, and when they will reach that senescence stage, what will happen is they will stop dividing. So the next question is, a cell line is what of a cell strain. So again, if you have listened to the lectures carefully, you'll know that a cell strain is basically a lineage of cells which are originating from the your primary culture. So your strain can either be divided from a primary culture or from a cell line. So here what has been asked in your question is, what is cell line then to a cell strain? So a cell line is going to be a superclass of your cell strain. Why is it so? It's so because the cell strain is going to be derived from a cell line, and a cell strain is different from a cell line in the sense that one cell line can give rise to, suppose this is some X cell line, so it can give rise to multiple cell strains. And these cell strains, these ABC cell strains, which have been derived from the same cell line, that is X, they can differ uh, in some genetic change, or they may have some specific property which is going to be specific only to the cell strain. So this is how a cell strain is different from a cell line, and a cell strain is derived from that cell line, and it has some specific characteristic which is going to be specific to that cell strain only. So the next question is, DMSO acts as a what does DMSO act as during cryopreservation of cells? So DMSO basically acts as a protective agent, and DMSO glycerol, these are very routinely used cryoprotective agents when we are doing cryopreservation of cells. So what happens in cryopreservation is, suppose you have your cells and like you go on culturing them, but your experiment has stopped. Now what you want to do, you want to preserve your cell, suppose because you want to start another experiment 1 month from now. So it's not feasible to go on keep on culturing them because again, since they are mostly finite cell lines, they are going to lose rigor, they are going to lose vitality. So a prudent option is to preserve those cells. So this is your cell line and you wish to preserve them. So the most commonly used method, or the widely used method for preserving cells, is cryopreservation. So here what happens is, what we do, so first what we do is we take our cell line. So we have some petri dish or a flask where cells are growing. From there we get those cells out, that is we detach the cells. I'm talking about adherent cell lines. So here what we do, we detach the cells, and once we have detached the cells, so now we can collect them in a tube. Suppose you collect your cells in a tube. So here what you can do is you can add your uh, cryo protective agent, or CPA as it is commonly called. This cryoprotective agent can either be glycerol or DMSO. Now, what this cryoprotective agent does is that it prevents ice crystal formation. So, what happens is, like your cells, your cells will have water inside. Uh, so, like when you are going to, so as the name suggests, cryopreservation. Cryo means cold. So you are going to store your cells at a cold temperature. So here what will happen, since the cells have water, like as soon as you're going to put them in a cold temperature or sub-zero temperatures, what will happen is these water molecules are going to freeze and they're going to be converted to ice. Now just imagine having ice inside your cell. So what will happen is your cells will burst. So we add these cryoprotective agents or CPA, that is glycerol or DMSO, to prevent this ice crystal formation. So this is an image which explains how your cryoprotective agent or how your DMSO is preventing the ice crystal formation. So here what is happening? Here we have our cell, and this cell is suspended in physiological solution, like it is in a normal culture environment. So normal culture environment means it is at 37 degree. So this is the temperature at which most of the cells grow. So in a normal environment at normal osmolarity, this is how your cell is going to look if you think about the water molecules which are inside the cell. So these are, as you can see, these are the water molecules inside the cell. Now what will happen is when you add the cryoprotectant, like, and another important point which should be mentioned here is that cryoprotectant is usually added at a controlled temperature, some like at 4 degrees Celsius or a temperature like that. So when you add your cryoprotectant outside, like in the media, what will happen is this medium of a normal osmolarity will change to a hypotonic extracellular solution. So after that, once you have added your cryoprotectant, what will happen is because of this hypotonic solution, what will happen is like your water and DMSO will get exchanged, and your DMSO, so this is the cryoprotectant CPA molecule. So this molecule DMSO will get inside the cell, and once it gets inside the cell, what will happen is it will not, because of this exchange, it will not allow the formation of the ice crystals. So once this is done, you can store your cells at liquid nitrogen, and the temperature of liquid nitrogen is minus 190 degree minus 196 degrees Celsius. So this is how your DMSO or glycerol or any cryoprotective agent works and prevents the formation of ice crystals, because if ice crystals will be formed, your cell will no longer be viable. It won't be able to grow again. So the next question, can everyone please mute them their mics? There's a lot of disturbance in the background. So the next question is, temperatures for cryopreservation are in the range. Again, we have different options. The first option is more than minus 130 degrees Celsius. The second option is more than 0 degrees Celsius. The third option is less than minus 130 degrees Celsius. And the fourth option is less than 0 degrees Celsius. So as I discussed earlier, cryopreservation means preservation at cold temperature, and so the temperature is obviously going to be less than 0 degree. And as I mentioned earlier, like cells are mostly stored in liquid nitrogen. So and the temperature of liquid nitrogen is minus 196 degrees Celsius. So your So here the correct option is the temperature for cryopreservation will be in the range of somewhere less than minus 130 degrees Celsius, because minus 196 degrees Celsius is less than minus 130 degree. So very briefly, I'll explain to you the method of cryopreservation, like how cryopreservation is done, what are the steps, and why is it important to gradually go from higher temperature to these sub-zero temperature and finally to this temperature of liquid nitrogen. So at first your cells are at physiological temperature, that is your cells are growing at 37 degrees Celsius. So
Ma'am, your screen not visible.
sorry, can everyone please mute your mic? Okay. So here your cells are growing at a physiological temperature, that is 37 degrees Celsius. So after that, what happens is you cool down those cells and you cool them down to somewhere around minus 5 degrees Celsius. And here what is happening is you have added your CPA. CPA will be your cryoprotective agent, which let us assume in this case is DMSO. So here we have added our DMSO, and then we have cooled the temperature of the cells to around minus 5 degrees Celsius. So here what will happen is this like slowly, because the osmolarity of the extracellular environment has changed, as I explained in the previous slide. So like your CPA molecules will get exchanged with the water which is present inside the cell. And like now here what will happen is, if you are not gradually reducing the temperature, suppose directly from 25 degrees Celsius or 37 degrees Celsius, if you are going directly to minus 196 degrees Celsius, then what will happen is uh, like your CPA will not get the time to percolate inside your cell, and this instant instant rapid cooling may cause the death of your cell. That is why instead of directly taking the cells from 37 degree and directly keeping them in liquid nitrogen, what we do is that we gradually reduce the temperature. So here what you are seeing is gradual reduction in temperature. Because see, the whole purpose of cryopreservation is to ensure that when you are going to revive those cells and when you are going to culture those cells again, your cells should have vitality. Like they should not be dead. You want to culture them again. So if you do not do this gradual reduction in temperature, then what will happen is your cells, when you culture them again, they will be dead, because the cryoprotective agent will not get ample time or temperature to work. And that is because what will happen at minus 196 degrees Celsius is that, like, there will be no thermodynamic reaction, like nothing will be happening. So obviously this exchange between the CPA and the water molecules is also not going to happen. So what you want to do is that you want, like, once you have added the CPA, you want to reduce the temperature, but you also want to reduce the temperature in such a manner that like this exchange between CPA and water molecule happens. So for that, what we do, like usually what we do, from 37 we go to somewhere around 22 to 25. From there we go to 4 degrees Celsius, then 0 degrees Celsius, then -5, -20, -30, or then -80. Then we put our cells, preserved cells, in -196 degrees Celsius in liquid nitrogen. And here another important point which should be mentioned here is that in liquid nitrogen, so there are two phases. First is the vapor phase, and second is the liquid phase. So preferably we keep the cells in the vapor phase, and we do not prefer to keep the cells in the liquid phase. So moving on to the next question. So the next question is, which of the following is a non-adherent cell type? So the first is skeletal muscle cells, second is fibroblast, third is cardiomyocytes, and fourth is red blood cells. So for this question, Professor Manak Das discussed it in very detail in his lecture, that there are two different kind of cell types. First is adherent and the second is non-adherent. So adherent will be your cardiomyocytes, fibroblasts, as well as your skeletal muscle cells. But your non-adherent cells will be all your blood cells, and so the answer is red blood cells. And the reason they are non-adherent is they have to do gaseous exchange, they have to carry oxygen, and if they become adherent, then like our life process will stop. So the non-adherent cell type is the RBCs, or the red blood cells. Next question is, anaerobic respiration occurs in, first is presence of oxygen, second is absence of oxygen, third is presence of hydrogen, fourth is absence of hydrogen. So here, as the name suggests, anaerobic, something which is not aerobic. Aerobic for us means oxygen should be present. So anaerobic means oxygen will be absent. So anaerobic respiration occurs in the absence of oxygen. Can everyone please mute? There's a lot of disturbance going on in the background. So moving on to the next question. So the next question is, if a terminally differentiated cell type gains stemness, what is it called? So if a cell which has already differentiated, if it again it gains stemness and again it's going to differentiate, then it is called de-differentiation. This point of differentiation and de-differentiation was also discussed in detail in the lecture, like in the video lecture. And the next question is, like, this is the cell's ability to differentiate into multiple cell types. So a cell's ability to differentiate into multiple cell types will be its potency. So depending on that, the cells can be multipotent or pluripotent. So I went to the discussion forum and there were a lot of questions and lot of doubts about the ECM or the extracellular matrix. So a term which is cementing material was used in the beginning lecture, I think, and there was a lot of confusion in the discussion forum about the cementing material and the ECM and whether these two are the same. So cementing material is not a terminology. The correct terminology is ECM. So I assume there's a lot of doubt regarding ECM. So very briefly, I'll explain what ECM is. So ECM or extracellular matrix. As the name suggests, it is extracellular. So it is not inside the cells, it is outside the cells. So ECM is an integrate network which is composed of different kinds of macromolecules and which are organized in a cell or tissue specific manner. So the different kind of macromolecules that we are talking about here can be collagens, proteoglycans, elastin, or different cell binding glycoproteins. So the ECM is actually a very complex network, and uh, till date it has not been possible to know all the components and all the macromolecules and everything that is involved in organization of the ECM, and that is because like ECM is complex and different kinds of signaling pathways and different kinds of signaling molecules, everything which is interlinked together is involved in the ECM. So the main function of ECM is to provide a stable structure to cells or to tissues, and the ECM majorly contributes to the mechanical property of tissue. So in this schematic, like I added this schematic so that you can appreciate that both cells and the ECM are interlinked. So what happens is all the components of like making this ECM are secreted by our cells only. It's not like just because it is extracellular matrix, the components are coming from somewhere outside the cell and then providing an environment to the cells to adhere to or attach to. No, it's not so. What happens is this matrix is secreted by the cells only, and once it is secreted by the cells, what happens is by matrix remodeling, uh, the extracellular matrix is formed, and once the extracellular matrix is formed, what happens is like again it will signal cells, a similar kind of cells which can be in one particular kind of extracellular matrix, and then what will happen is like the those kind of cells or those kind of cells which are there in that particular kind of extracellular matrix, they will be present in that ECM. So these two are not exclusive, they are actually inclusive of each other, and it's kind of a feedback mechanism where both the cells and the extracellular matrix are interdependent upon each other. And there was one more doubt about like what happens when cells become cancerous. Like does it like can this survive in any kind of extracellular matrix, because they are kind of defying their own ECM. So what happens then? So the answer is not as clear, like it's not as simple that whether if a cell is becoming cancerous, then like suppose if we have a cancer cell. So the question is, can it survive in any kind, like kind of ECM. So it will mostly depend on what kind of cancer cell it is. And when I say what kind of cancer cell, I mean it will depend on what kind of markers there are present on the cancer cells, and depending on that, these markers will decide like where in the like in which kind of ECM the cancer cell is going to survive. So like a broad answer will be no. It's not like any cancer cell can survive in any kind of ECM, but this promiscuity to a particular kind of ECM is what helps in the metastasis of cancer cells. So there are some more and some additional questions which I'd like to discuss so that your understanding of the week 1 lectures become more clear. So the first is on differentiation, the function of cells become less specialized, more specialized, it's not known, or has no correlation to specialization. So what happens on differentiation is your cell functions becomes more specific, because once your cells have differentiated into a particular kind of tissue, its function is going to be more specific. Next is, what is actin? Is actin a sugar, a lipid, a carbohydrate, or a protein? So the correct answer is actin is a protein, and like this actin and myosin were discussed in one of the video lectures, so you can again refer to that. The next question is, which of the following proteins is responsible for force generation in skeletal muscle? So again, there are four different options. First is keratin, second is myosin, the third is collagen, and the fourth is insulin. So the protein which is responsible for force generation in skeletal muscle is myosin. So this is basically all for today's session. I hope your doubts got clear, and this session, like the recording of this session, will be uploaded on YouTube, and that link will be shared with you. So if again still you have any doubts, what you can do is you can post your questions there, or you can post your questions in the discussion forum in the NPTEL group, and there I'll be like happy to help you out. Thank you.